Optimization of lipase activity obtained from dairy industry wastes. Abstract optimization of media components is very essential for the production of lipase to enhance its activity in order to meet wide variety of industrial applications. The enzyme production by the microorganisms in the environment is very low, hence it is essential to improve under required optimum growth conditions. This study mainly focuses on the enhancement of lipase production and its activity, which includes the environmental, nutritional, and biochemical factors of the media. In order to accomplish the enhanced activity, media parameters such as lipid and carbon and nitrogen sources along with other process parameters were altered. The lipase production mainly depends on nitrogen source, carbon source, metal ions, pH, and temperature and incubation time interval for media enrichment. The observations includes the screening of lipolytic bacteria by quantitative estimation of lipase by titrimetric method for isolated bacteria from dairy industry waste, confirmed on tributyrin agar and found to be highest lipase enzyme producer at an optimum pH 8, temperature 370 C with sunflower oil as substrate, whereas the nitrogen and carbon sources found to be peptone and glucose respectively. Keywords: bacteria, tributyrin agar, lipolysis, enrichment. Introduction. Dairy industries around the world produces wide range of products like cheese, milk, buttermilk, etc. During the manufacturing of these products, the industries are involved in the production of solid and liquid wastes. The effluents which were collected from the dairy industry consist of wide range of proteins, fats, and possibly other active substances. Microbiota are adapted to the wastewater physiochemical conditions and produces wide range of enzymes like lipases, proteases, etc. Some new active bacterial strains may be identified in wastewater effluents from dairy industries. Lipases are ubiquitous enzymes which are found in animals, plants, fungi, bacteria. Enzymes from microbial source are more stable than their corresponding plant and animal enzymes, and the production is more convenient and safer. Microbial lipases have been produced by yeasts, fungi, and bacteria as extracellular, intracellular, and cell-bound enzyme. It has been estimated that only about 2% of the world's microorganisms have been tested as source of enzymes, Sahu et al. 2011. These enzymes are generally involved in increasing free fatty acid FFA concentration through the hydrolysis of fats or lipids. They are mainly involved in hydrolysis, which catalyses the breakdown of triacylglycerol to glycerol and fatty acids. Hence these enzymes are used to catalyze interesterification of fats and oils to modify glycerides. Lipase production is also used as a marker for pathogenicity in some medically important bacteria. Many varieties of lipases are produced from both gram-positive and gram-negative bacteria. The process of lipolysis is to release free fatty acids FFA and partial glycerides which have either detrimental or beneficial effects in the environment. Lipolytic enzymes have wide range of industrial applications in food, dairy, paper, textile, leather, and detergent industries. These lipolytic enzymes are also used in wastewater treatment, production of fine chemicals, pharmaceuticals, and cosmetics, synthesis of surfactants and polymers, vegetable fermentation, and meat curing. Due to the wide range of industrial applications, lipases obtained from microorganisms are more interesting as they can be produced with better yields. There are many varieties of catalytic activities that can be used in several applications, and the genetic manipulations are easily available. The enzymes are heat stable and involved in the spoilage of various dairy products, Subhamvarma et al. 2014. 2. Materials and methods. 2.1 Collection of dairy wastewater samples. Samples from dairy industry wastes were collected in a sterile container and stored at 4 degrees C until the analysis was carried out according to the standard methods of APHA, American Public Health Association, and Tribty and Goyal, 1984. All experiments in the study were carried out with isolated lipolytic bacteria in triplicates, and the averages of the results were displayed. In the present study, dairy wastewater samples were collected and screened for the presence of lipolytic bacteria. 2.2 Isolation of lipolytic bacteria. Selective isolation of lipolytic bacteria from dairy wastewater samples were performed by using olive oil emulsion medium in screening medium for primary isolation and later confirmed on tributyrin agar medium. 2.2.1 Preparation of enrichment media. Enrichment media is prepared by using peptone, 0.05% with V, KH2PO4, 0.15% with V, Na2H2PO4, 0.1% with V, MgSO4, 0.05% with V, NH4, 2SO4, 0.5% with V, NaCl, 0.5% with V, olive oil emulsion, 1% V/V was prepared by mixing with 1 volume of the olive oil with 4 volumes of 5% polyvinyl alcohol, PVA. This emulsified solution was kept in orbital shaker at 250 rpm for overnight at 37 OC. Then the prepared oil emulsion was autoclaved at 121 OC for 20 minutes at 15 pounds pressure and the P was adjusted to 7.0. Then 5 ml of each sample was added to 50 ml of enrichment media in 150 ml flasks. The flasks were incubated on a shaker at 200 rpm at 37 OC for 72 hours. 2.2.3 Preparation of confirmatory media. Further to confirm the lipolytic isolates, the tributyrin broth medium was used as confirmatory media. This medium consists of components like tributyrin, 1% V/V, yeast extract, 0.3% with V, peptone, 0.5% with V. 2.3 Screening of lipolytic isolates. 2.3.1 Primary screening of lipase-producing bacteria. 5 ml of each sample collected from dairy industry wastes were suspended in 100 ml of enrichment media and agitated in orbital shaker incubator at 37 OC at 200 rpm. The enriched suspensions were used to make the dilutions from 10 to 1 to 10 to 10 by serial dilution method. 2.3.2 Confirmation of lipolytic bacteria. Lipolytic bacterial colonies were screened by qualitative plate assay method in the presence of tributyrin agar medium for 72 hours. Zone of clearance was observed due to hydrolysis of substrate tributyrin because of extracellular lipase production by lipolytic bacteria. 3. Lipase activity determination. The better clearance zone formed colony was selected for further studies. Qualitative test was done by hydrolysing the medium to form a clear zone and quantitatively by using titrimetric method. Lipolytic activity was checked quantitatively by titrimetric method. 3.1 Estimation of lipase activity in the lipolytic bacteria. Extracellular lipase assay was done with crude enzyme which is collected by centrifuging the bacterial culture for 10 minutes. 1 ml of the crude enzyme is mixed with 4 ml of olive oil emulsion and 5 ml of 0.5 m phosphate buffer at pH 7. Then this mixture was incubated for 15 minutes at 37 degrees Celsius. After incubation, reaction was terminated by the addition of 2 ml of acetone, ethanol mixture, 1:1. Then 2 drops of phenolphthalein indicator, PP, was added and titrated against 0.1 NaOH until the color changes to pale pink. Lipase activity can be calculated by the following equation: 1 lipase unit, U, is defined as the amount of lipids required per minute to liberate 1 mole of fatty acids. In the current study, lipase activity is measured in the presence of olive oil as the substrate. Lipase activity, U/ml, equals, AB, xn, NaOH, x 1000, /VE, 1. Description. A equals volume, ml, of NaOH for titration of the sample, B equals volume, ml, NaOH for blank titration 1000 equals conversion to mole, VE equals total volume of the mixture of crude extract. 4. Optimization of the conditions for lipase activity. The media parameters should be altered in order to optimize the lipase activity. For media optimization, the following parameters are considered, i.e., varying pH, from 5 to 9, varying temperature, from 270 C 300 C 330 C 370 C 390 C 420 C, lipid sources, palm oil, mustard oil, sunflower oil, coconut oil, tween 20 and butter, carbon sources, glucose, fructose, galactose, and sucrose, nitrogen sources, peptone, yeast extract, beef extract and sodium nitrate. The activity of lipase enzyme from isolated bacteria present in dairy industry wastes was optimized by considering the effect of following conditions. A. Effect of pH on lipase activity. The optimum pH for maximum lipase activity for the isolated bacteria was determined by considering the effect of different pH in the tributyrin broth ranging from pH 5, pH 6, pH 7, pH 8, pH 9, whereas the other parameters remain unaltered. B. Effect of temperature on lipase activity. To select the optimum temperature for the maximum lipase activity bacteria, the effect of temperatures was studied ranging from 270 C, 300 C, 330 C, 370 C, 390 C, 420 C by keeping the remaining parameters unaltered. C. Effect of different carbon sources on lipase activity. In order to optimize the activity of lipase obtained from isolated bacteria, different carbon sources like glucose, fructose, galactose, sucrose were added to enrichment broth at a final concentration of 1%, with V, whereas other parameters remain unaltered. D. Effect of different nitrogen sources on lipase activity. To optimize the activity of lipase obtained from isolated bacteria, different nitrogen sources like yeast extract, sodium nitrate, beef extract and peptone were added to the enrichment broth at a final concentration of 1%, with V, whereas other parameters remain unaltered. E. Effect of different oils as lipid source on lipase activity. Lipid sources for the production of lipase was optimized by replacing the olive oil present in the enrichment media with different oils like palm oil, coconut oil, sunflower oil, mustard oil, tween 20, butter at a final concentration of 1%, with V, and whereas other parameters remain unaltered. F. Effect of different metal ions on lipase activity. In order to optimize the activity of lipase obtained from isolated bacteria of dairy industry wastes, different metal ions like Mg plus 2 Na plus K plus Ca plus 2 Zn plus 2. Fe plus 2 were added at 1 mm concentration and incubated for 72 hours. Finally Mg plus 2 Na plus were tested at different concentrations 1 mm, 2 mm, 5 mm, 10 mm, and 20 mm and incubated again for 72 hours keeping the other parameters unaltered. 3. Result and discussion. 3.1 Optimization of media parameters. The media parameters should be optimized in order to enhance the lipase activity. For the media optimization the following parameters are considered, i.e., varying pH of 5, 6, 7, 8 and 9, varying temperature, from 270 C, 300 C, 330 C, 370 C, 390 C, 420 C, lipid sources like palm oil, mustard oil, sunflower oil, coconut oil, tween 20 butter. Carbon sources like glucose, fructose, galactose, and sucrose were used. Effect of Na plus and Mg plus 2 metal ions at different concentrations such as 1 mm, 2 mm, 5 mm, 10 mm, 20 mm were analyzed on lipase activity, after which the different nitrogen sources like peptone, yeast extract, beef extract and sodium nitrate were also considered. All the experiments were done in triplicates and the averages of each experiment is analyzed. A. Selection of pH optima for lipase activity. It is necessary to determine the pH optima of enzymes as the enzymes are sensitive to the pH of their reacting medium. The maximum efficiency of enzyme activity can be obtained only at pH 8 with different substrates like sunflower oil, mustard oil, butter, coconut oil, palm oil, and tween 20. The maximum lipase activity was found to be 42 IU/ml for sunflower oil at pH 8 as indicated in Fig 1. B. Selection of temperature optima for lipase activity. The enzyme activity is found to be optimum with all substrates at 37 C exhibited. However, the activity remains less at pH 6 and 7. The lipase activity was almost the same at all other temperatures taken under study and the optimum activity is observed with sunflower oil as substrate. The maximum lipase activity was found to be 30 IU/ml at 37 C as indicated in Fig 2. C. Selection of carbon sources for optimum lipase activity. For selection of optimum carbon source, different types of 1% carbon sources like glucose, fructose, galactose, sucrose were used in the media preparation in which the maximum peak activity can be seen only for glucose as carbon source with all different substrates. The maximum lipase activity was found to be 42 IU/ml as sunflower oil as substrate as indicated in Fig 3. D. Selection of nitrogen sources for optimum lipase activity. Generally, microorganisms produce high amounts of lipase when provided with organic nitrogen sources such as peptone and yeast extract as nitrogen sources for lipase production. In the present study, when inorganic nitrogen source such as sodium nitrate is used, but didn't show any lipase activity. Optimum activity is observed only with peptone when used as a nitrogen source and with sunflower oil as a substrate when compared to other substrates. At these conditions the maximum lipase activity was found to be 32 IU/ml as indicated in Fig 4. E. Selection of lipid sources as substrates for optimum lipase activity. For selection suitable substrates for optimum enzyme activity sunflower oil, palm oil, mustard oil, coconut oil, butter, and tween 20 were used as a source of substrates, in which the sunflower oil is found to be a better substrate with highest enzyme activity when compared to other substrates at 72 HRS of enrichment. Maximum lipase activity was found to be 46 IU/ml with sunflower oil at as indicated in Fig 5. Based on the results, lipase activity is enriched with the isolated bacteria in presence of glucose as carbon source, peptone as nitrogen source for 72 hours and activity is checked repeatedly in all conditions, where sunflower oil is found to be a best substrate at optimum pH 8 and at 370 C temperature. The average activity lipase activity was found to be 38.4 plus or minus 6.2 IU/ml. F. Effect of metal ion on lipase activity. For selection of suitable metal ions for optimum enzyme activity, different metal ions like Mg plus 2 Na plus K plus Ca plus 2 Zn plus 2. Fe plus 2 were checked for the optimum lipase activity, in which the maximum lipase activity was found to be for sodium and magnesium ions at 1 mm concentration after 72 hours of incubation. From which the two metal ions, that is Mg plus 2, Na plus, which has shown the maximum lipase activity, that is 10.9 U/ml and 14 U/ml respectively. Hence they were again tested at different concentrations: 1 mm, 2 mm, 5 mm, 10 mm, and 20 mm concentrations, in which the maximum activity was found to be 17.6 U/ml at 10 mm of Na plus and 15.7 U/ml at 20 mm of Mg plus 2 metal ion concentration, as shown in the Fig 6 and 7. Discussion. The results showed for optimization of media in order to enhance the activity was observed only after enrichment with sunflower oil for 72 hours, glucose as carbon source and peptone as a nitrogen source. Further, it is observed that the enzyme possesses optimum pH at 8, temperature at 370 C. Based on the results, we conclude that isolated bacteria from dairy industry wastes has optimum lipase activity with sunflower oil as substrate, with maximum activity at 46 IU/ml after 72 hours of incubation, whereas peptone as a nitrogen source showed maximum activity with 32 IU/ml and glucose as carbon source was shown maximum activity of 42 IU/ml. At optimum pH of 8 it showed 42 IU/ml, and at its optimum temperature 370 C maximum activity is found to be 30 IU/ml. Finally, the optimization for lipase activity is done repeatedly for several times at the optimal conditions, and the activity is calculated as 38.4 plus or minus 6.2 IU/ml. The activity of enzyme is very susceptible to media composition and other parameters. Formerly, lipase production from Aspergillus niger was acquired by solid substrate fermentation by from gingerly oil cake as substrate, Kamini et al. 1998, however, we observed sunflower oil as substrate, which has shown highest lipase activity in submerged fermentation process. Further, it was already investigated that lipase production from milk isolate Serratia rubidia and observed pH 7 to 8 and temperature 30 to 40 degrees Celsius, range optimum for maximum lipase production, Immanuel et al. 2007. Casein is selected as a nitrogen source, 6.5 plus or minus 0.015 U/ml, and soytone as a suitable substrate, 9.4 plus or minus 0.02 U/ml, for accelerating lipase production and activity for lipase obtained from Serratia rubidia, Immanuel et al. 2007. It was also noticed that the lipase production by Pseudomonas sps 34 was maximum with two nitrogen sources, namely tryptone and soytone. Even some of the studies reported that lipase production by Penicillium aurantiagrum was high when inorganic nitrogen source was used. Basically, organic nitrogens such as peptone and yeast extract, as suggested by our observations, have preferably been used as nitrogen source for lipase production by various Bacillus species, viz. Bacillus strain A31, B. alcolophilus, B. licheniformis strain H1, different Pseudomonas, viz. Pseudomonas sp, P. fragi, P. fluorescens BW 96 cubic centimeters, and Staphylococcus hemolyticus, OETAL, 1999, Gainum ETAL, 2000, Lancer ETAL, 2002, Sharma ETAL, 2002, while tryptone and yeast extract have been used for S. hemolyticus L62, OETAL, 1999. In our study, isolated bacteria shown similarity to all Bacillus sp. In the maximum production of lipase at 72 HR of enrichment, and similar results were examined by Sarkar ETAL, 1998. In contrast, 12H of the optimum incubation period was observed for A. calcoaceticusant Bacillus sp. Mahler ETAL, 2000, and 16H the optimum incubation period for B. thermoc Dannert ETAL, 1997. However, the maximum lipase activity was shown to be 72 and 96H in the case of P. fragi and P. fluorescens, respectively, Pabai ETAL, 1996, Dong ETAL, 1999. Optimum temperature for lipase production was observed with respect to optimum temperature for growth. In the case of B. subtilis Pxernel 39, it is observed the optimum temperature at 45 degrees Celsius, which is in agreement with the result of Olibers ETAL, 2017. Conclusion. Experimental results suggest that various media components influence the lipase activity by the isolated bacterial strain from the dairy industry waste. Optimization of media parameters for enhanced activity was observed at varying pH, temperature, carbon sources, nitrogen sources, and different substrates, which has shown significant effect on lipase activity. Based on the results, we concluded that isolated bacteria from dairy industry waste has optimum lipase activity with sunflower oil as substrate after 72 hours of enrichment, whereas peptone as nitrogen source and glucose as carbon source along with increased activity with metal ions like Na+ and Mg+2 at an optimum pH 8 and optimum temperature at 370 C. The maximum lipase activity was found to be 38.4 plus or minus 6.2 IU/ml.